Hello and welcome to our presentation. We are presenting our poster on a scoring statistic to measure periodic phasing patterns of piRNA biogenesis in mosquito small RNA transcriptomes. And this is being presented by myself, Aubrey Odom, and Neal Kewalramani and Nate Borders, and we're all students in the bioinformatics PhD program at BU. And we also have collaborators Nelson Lau and Gargi Dayama, and they work at the Boston University School of Medicine. Nate Borders will give us a little bit of background on our project.
Anopheles mosquitoes are carriers of malaria, one of the worst human pathogens, but they vector relatively few viruses. On the other hand, Culescine mosquitoes carry a whole host of human viruses, such as dengue and yellow fever, and this is largely due to their immunity to those viruses. One hypothesis as to how this immunity has arisen is in these characteristic piRNA phasing patterns that Culescine mosquitoes often display. On the left of the lower figure there, we can see a periodic curving pattern, which is a cutting pattern in the piRNA biogenesis. So each piRNA is cut in a very specific location. And then on the right, we can see that the Anopheline mosquito does not have a very strong characteristic piRNA phasing pattern. So one way to figure this out and find out which piRNAs are contributing to these phasing patterns is in a genome scale subsetting of the piRNA loci. However, there aren't any tools to grade these piRNA phasing patterns other than just by eye ranking and seeing how strong they are. So that is the objective of our project here. We're trying to develop a pipeline to sort piRNA curves based on phasing pattern strength. To do this, we extracted 7 different features from the curves, and we used them to cluster and then sort them into good and bad and okay phasing curves in terms of their phasing strength. And there was no one-size-fits-all metric, so we ended up using things such as the standard deviation between the different peaks. If it was lower, then we could say it's more periodic. There's also a variety of different metrics for determining the AUP, which is area under the peaks, and the smoothing error. So we smoothed the curves and sort of determined how much the curve deviates from the smooth curve. So all of these were required to give us metrics that we could cluster the curves using.
For subsetting the curves, we use a process called downsampling, where we can generate new curves based on the percentage of reads we want to extract. Here we looked at 80, 60, 40, and 20% of the reads. The datasets chosen optimize the quality of their curves, and the downsampled datasets were processed using the GATK pipeline from the Broad Institute. We then visually compared the resulting curves against the other curves produced, along with the original curve as well. In terms of our results, we first wanted to find the optimal number of clusters for this multidimensional dataset. We used the elbow method to find the number of clusters, and based on this method, we decided to use 8 clusters. Then from these clusters, 4 groups were made to classify the overall quality of the curves. Next, we classified the curves into groups based on which cluster was closest to the curve, and here are 2 example clusters. What we wanted to do next was determine how well the k-means algorithm worked in matching our by-eye ranking of these curves. To do this, we developed a training set of 265 curves, which was used to find the 8 clusters in the 4 groups, and a test set to generalize the results of the k-means algorithm. Each set was compared to a by-eye ranking of these curves using a Wilcoxon signed rank test. We found a p-value of 0.216 for the training set and a p-value of 0.275 for the test set. In terms of downsampling, here we have a GIF of the effects of downsampling. Specifically, this curve is from Aedes albopictus. What we notice is that many curves maintain their periodicity when the percentage of reads decreased, but the jaggedness of these curves increased as the percent of reads decreased.
So as far as what we can gain or glean from our results, we did find for the k-means clustering segment of our project that ranking by k-means clustering didn't seem to be statistically different from that of the by eye ranking based on our p-values being greater than our alpha of 0.05. So we concluded that k-means clustering can be used to rank curves accurately when compared to the by eye ranking. However, we do find that there is more data that is needed for a more precise method of ranking, such as a random forest classifier or some other method of machine learning. As far as the downsampling segment of our project went, we hoped to see with downsampling some point where the curves would lose a significant portion of their periodicity and smoothness. And if we found that point, then that would give us some evidence of piRNA clusters contributing more strongly to the curve's qualities. However, we did find that the deterioration didn't happen exactly as we expected it to. For 9 of the 15 of our curves, we found that rapid deterioration occurred even as they decreased to 80% of the reads out of 100%. And these curves became unidentifiable in the process as they declined from 100% to 80%. Any real curvature or smoothness seemed to disappear. And then for 6 out of 15 of our curves, which was the other subset, some increased jaggedness occurred as the reads decreased to 20%. The overall shape, including the number and position of the peaks, largely remained the same, however. And you can see that with the example curves that we showed previously. And unfortunately, we were unable to completely validate the hypothesis of piRNAs or a subset coming from one major genomic locus due to the rapid deterioration that we mentioned with 9 out of the 15 curves. So we're still uncertain of the source of that problem, and that is something that we'll be looking into in the future. As far as future goals, we are hoping to continue to work to discover the source of the piRNA phasing patterns. And one of our driving questions is a single population of piRNAs or a subset coming from one major genomic locus that accounts for most of the more highly regular, smooth phasing pattern. So one method by which we can go about doing this would be to construct subsets of piRNA cluster loci that could potentially be contributing to these curves' best qualities. And then we could reverse the scoring device to conduct previous population subset analysis and then compare the other populations of piRNAs in Aedes and Culex families to determine whether they are worse in their phasing patterns. So in conclusion, we expect that the quantitative scoring mechanism that we've developed will be interesting to the greater scientific community that is interested in piRNA biogenesis. So we sought with our project to contribute some further understanding to the investigation of these strong phasing pattern signals that seem to occur mostly in mosquitoes. And we believe that the classification of these species according to their curve smoothness and periodicity characteristics will facilitate the extension of this knowledge. So thank you for watching our presentation today, and please reach out if you have any questions in the chat. Thank you.